Hello, you are welcome. Imagine a world where a scientist can cut and edit DNA with precision by using certain tool, certain method, by using, for example, biological or molecular scissors, like this. Now, we discuss something about restriction enzymes. What is meant by restriction enzymes? Restriction enzymes are a group of enzymes found and discovered inside bacterial cytoplasm, where the bacteria use this type of enzymes to digest and attack phage DNA into small pieces. When bacteriophage attack bacteria, it inserts its DNA. The enzymes inside bacterial cytoplasm move forwards or directed towards this foreign DNA and cut it at certain recognition sites. So the restriction enzymes are derived from bacteria, and the bacteria are considered the inventor of genetic engineering. Okay, sometimes restriction enzymes are called endonucleases. Why? Because these enzymes, when cut and digest DNA into small pieces, it digests the DNA and the cut DNA at recognition sites within the DNA molecules, not from both ends, within. Okay? They cut within DNA molecules, not at the ends. What's the function of these restriction enzymes? They recognize and cut DNA of viruses that attack bacteria. Okay, so they restrict viral infections. Mechanism. What about the mechanism of these enzymes? Precisely how they work. Each restriction enzyme binds to a specific target site, restriction or recognition site on DNA, and it cuts at that site with precision. Look here. Yes, this restriction enzyme, okay, cut at restriction sites at certain sequence of DNA, foreign DNA. This sequence around 4 to 6 bases, only short bases, short length, 4 to 6 bases. Okay? Restriction enzymes cut DNA in two ways. The first way, okay, like cutting cheese with knife. It forms something called blunt ends. Straight cuts across the sugar phosphate backbone. Blunt ends. Okay. The second method, by forming sticky ends. These sticky ends staggered cutting or cuts, leaving short unpaired base sequence. Here single-stranded DNA. This called sticky ends or adhesive ends. Okay, adhesive ends. Sticky ends can form hydrogen bonds with complementary sequence on the other DNA fragments cut with the same restriction enzymes from any source, like from plasmid, for example. Okay. But must the same restriction enzymes. Restriction enzymes or restriction sites are palindromic. What is meant by palindromic? Means something like reading the word the same in both directions, like radar, like civic, and so on. Here, look, if we read the sequence from right to left, CACGATT, like from left to right, CACGATT, the same letters in both directions. Okay, read the same in both directions. This is the meaning of palindromic. Like this English words: civic, radar, madam, noon, mom, and so on. Now, how does bacteria protect its own DNA from cutting with its own restriction enzymes? Okay, both are found inside the same bacterium: cytoplasm, where DNA is spread in cytoplasm, okay, and also restriction enzymes found in cytoplasm. Why restriction enzymes doesn't attack bacterial DNA? This is big question. Bacterial DNA is protected from these enzymes by either what? The first one, bacteria add some chemical groups like methyl groups, CH3, at recognition sites. Okay, bacteria can modify its recognition sites by adding certain methyl groups on it. Like here the chromosomal, the bacterial chromosome, where certain group of enzymes can add methyl group at the same recognition sites for the same DNA sequences. Okay, this modification, chemical modification used by bacteria to mimic or to hide the recognition sites by the restriction enzymes. Okay, here bacterial DNA, where this methyl group added at the recognition sites, okay, like flag, like here lock, country's flag above embassy or building, to allow us or allow others to show that this is self, not foreign, okay, during war. Or second mechanism of bacteria, they uses what? They uses no restriction sites, no recognition sites found in their DNA content. So as no recognition sites, the restriction enzymes find no target to attack it. Okay, thanks for watching and the best wishes. Hello, you are welcome. Imagine a world where scientists can cut and edit DNA with precision by using certain tool or certain machine, like for example biological or molecular scissors, like this. Something interesting in our topic, in our video about restriction enzymes. What is meant by restriction enzymes? Restriction enzymes, group of enzymes found in bacteria, okay, where the bacteria use this restriction enzymes to cut foreign viral DNA into small pieces with functionless. Here, when bacteriophage attack bacteria, it inserts its DNA content inside bacterial cytoplasm. Restriction enzymes can attack this foreign DNA and cut this DNA into small pieces at certain sites called recognition sites. Okay, so the source of this restriction enzymes are bacteria, and the bacteria are considered the inventors of genetic engineering. Okay, sometimes restriction enzymes called endonucleases Why they are called by this name, endonucleases? Endonucleases means this type of enzymes can cut within DNA molecules, not at the ends. Okay, within DNA molecules, not at the ends. What's the function of this restriction enzymes? They recognize and cut DNA of viruses, okay, that attack bacteria. They restrict viral infections by this method. Mechanism. What's the mechanism of restriction enzymes? Each restriction enzyme binds to specific target site, recognition or restriction site on DNA, and cuts at that site. Here in this figure, look at this sequence of letters GAATTC, okay, on 5-3- direction, and from 5- to other 5-3- direction. Also the complementary base pairing for the first one. Each restriction enzyme, in this example restriction enzymes EcoR1, can cut between G and A, okay, above, and the other strand from other direction G and A. So it leaves certain shape called sticky or cohesive ends or adhesive ends. Okay, this part can adhere with some complementary base pairing cut with the same enzyme. Look here, restriction sites are specific sequences, short sequences of DNA, range from 4 up to 6 bases. Here 6 bases in this example. Okay, restriction enzymes cut DNA in 2 ways. The first shape, by forming certain ends called plant ends. Here restriction enzyme BVU2, okay, this restriction enzymes cut both strands of DNA straight across, producing plant ends. Here plant ends straight cut across the sugar phosphate backbone of DNA. Okay, this is the first type. Second way, by forming something called sticky ends. The sticky ends target cuts, leaving short unpaired base sequence. Okay, single-stranded DNA, sticky adhesive ends. Okay, sticky ends can form hydrogen bonds with complementary sequences on the other DNA fragments cut with the same restriction enzyme. Must cut with the same restriction enzymes, like plasmid, for example. When we cut plasmid, bacterial plasmid, by the same restriction enzyme of HEND3, for example, in this part, and mix both sources of DNA, they can join together by certain type of enzyme called ligase enzyme. Okay, look, restriction sites are palindromic. What is meant by palindromic? Read the same in both direction. It means reading the word the same in both direction, from left to right and from right to left, like level, civic, mom, dad. If we read these words from left to right, is the same like from right to left. Okay, look here, palindromic, this sequence of DNA, okay, when recognitions where a restriction enzyme can recognize this a piece of nucleotides, it cut here between C and G and leave certain sticky ends. Okay, look, from right to left, above CA, CG, ATT, From left to right, CACGATT, the same. Okay, this is called palindromic sequence. Palindrome words like dad, pop, mom, level, none, and so on. Now, how does bacteria protect its own DNA from cutting with its own restriction enzymes? This big question. How the bacteria solves this problem? How it protects its DNA content, its chromosome, its circular DNA from attack with its own restriction enzymes? How? The bacteria uses two methods, two mechanisms. Bacterial DNA is protected from these enzymes by either the first method, by chemical modification. Bacteria add some chemical groups like methyl groups, CH3 groups at recognition sites. Okay, like here, this DNA, okay, circular DNA of bacteria, the main chromosome, okay, certain type of enzyme called methylated or methylation enzymes can add methyl group at the same recognition sites, so it mimics or hide the attack of restriction site by restriction enzymes. Okay, it hides the restriction sites. Look here, bacterial DNA, by adding methyl group on both recognition sites above and below on complementary strands. Okay, recognition sites now are considered methylated, so it looks like when we put our own country's flag above certain embassy during war. This means for our soldiers that is ours, this self-building, okay, not attacked by weapons, for example. Okay, here like country's flag, the same concept to show that it's own, like the country's flag above embassy or above certain building. Or another method, the bacteria use no restriction sites, no recognition sites in their DNA content, so not attacked by restriction enzymes. Okay, it uses both methods. Thank you for watching. See you next time. Have a nice day. Bye bye.